In this video, we will discuss about this RIBOmap, or we can say we will continue our discussion about RIBOmap, and we will discuss the specificity of the RIBOmap and its comparison with another mRNA translation system that is published in the past is that is STARmap. So let us start with the specificity of the RIBOmap. In this, what they used is they use mRNA that has this sequence, that is ACTB sequence, for translation. Simultaneously, they use two negative control, that is this MALAT1 and VTRNA1-1. So these are the non-translating RNA that has this gene sequence, and along with that they use the mRNA that can be transcribed that has this gene sequence ACTB. So when they did the experiment with the RIBOmap and also with the STARmap, they can find that for this RIBOmap the signals are positive only for this translating mRNA with this ACTB gene, whereas for other, that is MALAT1 and VTRNA1-1, they did not see any significant result. Whereas when they use this STARmap, their data shows that it can still present signals for MALAT1 and VTRNA1-1. So this indicates that their RIBOmap will predict more efficiently about the translating mRNA compared to the another published method, that is STARmap. And this thing has been quantified in this panel. In this we can see that the ACTB gene is translating is ACTB gene signal is very high, and is comparison to this MALAT1 and VTRNA1-1. However, we see some signals even in STARmap, whereas these are the non-translating, non-coding RNA. So later on, what they did is they used more stringent method in which they used a RNA and then inhibit the mRNA translation through this Huntington treatment. So, and then they use three different probes. First probe is that is 16 nucleotide before this this stuck of this mRNA by this Huntington treatment, and second probe is after the 100 nucleotides, around approximately 110 nucleotides after this Huntington treatment stop the mRNA translation, and this probe 3 is after around 360 nucleotides after this Huntington treatment stop the translation or stuck the mRNA translation. And then they check all the three probes. So without drug treatment, since complete mRNA will be translated, they see significant signals without drug treatment or without Huntington treatment. Later on, when they did the Huntington 9 treatment, they can see the high signals for the probe that is been before the that is 16 nucleotide before the stuck of this mRNA translation will show a high signal, whereas for this probe 2 and probe 3 we see some signals, however it is quite low. This further shows the specificity of the RIBOmap, that it can more prominently shows the signals only for the translating mRNA, and compared to the for the translating mRNA, and whereas the signals for non-translating part of mRNA is very low. And this thing is been quantified, this image has been quantified in this figure. We can see the signals for this probe 1 is very high, significantly higher than the probe 2 and probe 3. Later on, what they did is they compare this STARmap and mRNA through the in vitro translated by transfection, this lipofectamine transfection of in vitro translated mRNA. And when they transfect the RNA, there is a vesicle, and so some of the vesicles get translated. As it can see, this it binds to the ribosome, whereas some of the vesicles does not get translated. It is still present in the lipofectamine vesicles. So what they see is that in the STARmap results, the lipofectamine vesicles will also show signals, as we can see in the firefly signals and renilla signals. And in the merge file, we can see the signals of this intra vesicles mRNA. So this shows the non-translating mRNA also. The STARmap shows the non-translating mRNA signals also. Whereas when we use this, when they use the RIBOmap for analyzing this samples, they did not see that uh, intravesical mRNA or non-coding mRNA in their signal. And this can be and this thing can be seen in this merge file also. Whenever they use the STARmap, they can see the intravesical non-translating mRNA signal, and when they use the RIBOmap, they do not see intravesical non non-translating mRNA signal. So this figure made the point that their RIBOmap is far more robust and more specific towards translating mRNA, and it's better than the previously published method for uh, measuring translating mRNA, that is STARmap. If you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.